Good afternoon, everyone, and welcome to this EWMA webinar on bacteria and wounds, a better understanding for treatment. And today, apart from me, we also will hear from Marvin Whiteley from the United States, talk about gene expression of bacteria and wounds and infections, and also Frederik Hertz here from Denmark, who will talk about some information on how to use, not just to use antibiotics for treating wounds. And the aim of this program is to promote infection and prevention and control, correct diagnosis, and then also appropriate use of antimicrobials and antibiotics. And for the session, we also have these sponsors, which we are of course grateful for sponsoring EWMA and this session. And with this, I will initiate the session, talk about biofilms in wounds, what we know, and also maybe what we do not know. First of all, it's a question we have had, I think also Marvin had for many years. Why is it called biofilms? And the reason is that Bill Costerton, who was a basic researcher, an environmental biologist, he was together with some of his colleagues looking into recurrent infection of a patient with a pacemaker, and the patient had recurrent infections of Staphylococcus aureus. And when they extracted the pacemaker core, the pacemaker leads, they saw, as you see here in this scanning electron microscopy pictures, they saw Staphylococcus aureus attached to the surface. And Bill Costerton here has been examining rocks and surfaces in alpine creeks in Canada. And here in these alpine creeks, he saw that the slimy surface on the rocks and so on was this biomaterial and bacteria embedded in it. So he had termed that biofilms. And when he saw the bacteria on the surfaces also here, he immediately made the correlation that that was also the cause of infection, and that's why it's called a biofilm. And it's from his the core study or core paper, which really coined the term biofilm from 1982, and coined the relationship between clinical microbiology and clinical infections and biofilms. But also in 1920, there was this notion of this citation: there's always a well-known solution to every human problem. It's neat, it's plausible, and it's wrong. So I will talk a little bit about maybe why attached bacteria and biofilms are not the complete cause of chronic non-healing infections. And I think also Marvin will expand a bit on this. So when I started my career, I started to look into the chronic infection of cystic fibrosis patients, a chronic lung infection. And we detected, as you can see here in this microscopy picture, we detected Pseudomonas aeruginosa in red in big aggregates surrounded by polymorph nucleoleucocytes here in blue. And these biofilms were situated in all the major airways, as you can see in this explanted lung, and all these green pus pouring out of the major airways is the mucus of the CF patients together with all the bacteria and all the inflammatory cells. And these pictures are from a male who had cystic fibrosis and had the chronic Pseudomonas aeruginosa lung infection for more than 28 years. And in these 28 years, he received more than 1 kilo of topramycin, more than 10 kilo of beta-lactam antibiotics, more than 1 kilo of colistin, and a lot of inflammatory cells. But despite of that, the bacteria still persist. And here back in 2008, 2009, I really focused on the aggregated bacteria. But you can see actually in some of these surrounding small red dots, it's not everything which was in biofilms. We also focused a lot in the beginning on biofilms in chronic wounds, and also here we could detect both Pseudomonas aeruginosa and Staphylococcus aureus and so on aggregated. And this still is one of the best reviews on biofilms in chronic infections by David Lebeau, really going through device-related infections and tissue-related infections, and I really encourage you all to read this. But then also together with Marvin Whiteley, I'll present next, we started to think about is this termination of acute infections caused by planktonic bacteria and chronic infections caused by biofilms, is this really how we can divide everything? And we know from a lot of studies that, I have to go on back, that we can find these aggregates of Pseudomonas aeruginosa and other bacteria in the CF lung. But you can see there's also a lot of single cells. And so we took pictures of microscopy pictures of a lot of sputum samples and lung samples from CF patients. And then together with Marvin, we made these distributions of the biomass, and you can see more than 50% of the bacteria in these lungs were actually situated as single cells or extremely small microcolonies of 1, 2, 3, 5 bacteria. It was actually a smaller proportion than 50% of the bacteria which was situated in these large aggregates. And also when we look at the chronic wounds, so back in 2000 and from 5 until 10, we really had to focus on only to look at bacterial biofilms. So every time when I was looking in the microscope, when there were single cells, I just turned to another field of view because single cells we thought was just artifacts. When we look closer in the samples now, we can see a lot of single cells. And actually also there's this systematic review finding biofilms in acute wounds. And when we look in the wounds again, we can see a lot of the biofilms, but we also see a lot of single cells. So acute and chronic wounds contain both single cells and biofilms, and so we could also see in the CF lung. But another hallmark of clinical microbiology and microbiology within infections was, of course, founded with Robert Koch, and for him, and a statement which still today is the hallmark of clinical microbiology: the pure culture is the foundation for all research on infectious diseases. And also, if any of you have tried to grow bacteria in the laboratory, everyone, and also so we have done so, have really, really focused on the exponential phase of bacterial growth, really determine reproducibility and so on. So what we actually have done is that we have made all our model systems kind of a all-inclusive holiday for the bacteria. We treat them with all the nutrients and all the available carbon sources and so on, so they live a happy life, and all our experiments can be reproduced, and that's also what the journals demand. But bacteria out in the environment, they don't get an all-inclusive, and the bacteria in chronic wounds do not get an all-inclusive, because here they have to fight against predators and in the wounds immune defense and so on. So when we started to make model systems for biofilms, we have grown them on surfaces, reproducible mushroom structures, we have grown them in microtiter plates and so on, but all focusing on growing bacteria reproducible in an all-inclusive mode, not as they grow in the patients and in the environment. And also Marvin will touch upon this in his talk. So basically, what we have done is in clinical microbiology and in infections research is that we have had a sole focus on the bacteria attached to surfaces or suspended aggregates. We haven't really thought anything about there's a human body surrounding the bacteria, and there's all the host material. This is never really put into play when we culture bacteria in the laboratory. And of course, we have the extreme good mental model of biofilm growth from a reversible attachment to maturation and dispersion. But this is not how the bacteria grow in a laboratory. But we still grow bacteria in the laboratory in shaking cultures and microtiter plates and flow cells and so on. But if we take a step back, the way we grow bacteria in the laboratory and how we handle bacteria is we reduce complexity, and I think also we reduce relevance. It is almost as studying lions in cages, and we think we can understand the lions in Africa in the savannah, or birds in cages, and think we can study the birds out in the fields. So we really have reduced complexity, so now we can repeat and we have reproduced results. But I suggest here that also led to that we do not really understand how the bacteria are growing in infections. And that is very, very evident from the current treatment and also susceptibility testing in a clinical microbiology lab, because this is guided by the UCAST guided treatment, the minimal inhibitory concentration. This is a very well-defined and standardized method, and it will give a generalized antibiotic susceptibility framework, which the clinicians, of course, can use. But we see very, very often that the laboratory-guided minimal inhibitory concentrations still fail to treat especially chronic infections. And that is because they are optimized for reproducibility in the lab conditions. We have given them the all-inclusive mode, where we rather should have maybe tried to mimic more in vivo conditions. But we know that also in minimal biofilm eradication conditions and inhibitions is not equal to in vivo susceptibility. And the consequences of this is that we often see treatment failure, infections relapses, and also have a hard time diagnosing, especially bacterial chronic infections. So we propose that we need to understand the metabolotype, and that is the readiness or and how a bacteria will react in a given environment. For example, in the CF lung or in the chronic wound, we have to understand which genes is turned on and which pathways is being used, and that is also something that Marvin is studying. An example of this is this study from Louise Kruse Jensen and Mads Bue. Louise, she's developed this pig model for implant-related infections, where they drill a hole in the tibia of a pig, and then they insert a steel implant, and then they can add bacteria and so on to mimic an infection. And in this particular study, Louise mixed Staphylococcus aureus on the lab bench together with increasing amounts of gentamicin from 16 times MIC to 16,000 times MIC. And if these cultures have been left at room temperature or at 37 degrees overnight in a lab, all the bacteria would have been dead. But Louise, together with the implant, injected this solution, and then days later, she extracted the implant, and even in the highest concentration of gentamicin, there was still bacteria in one of the pigs. So something completely changes when bacteria enter not only the human body, but also here the pig body. And this is what we have started to term the infectious microenvironment, that the microenvironment around the bacteria, especially in chronic infections, is completely different from what we normally see in the human body. And infectious microenvironment can be caused by surgical incisions, insertion of implants, edemas, thick mucus in CF, and so on. And it will lead to a changed microenvironment. and this disrupts homeostasis and a lot of pathology. And the microenvironment will then be characterized by changed physical factors, products of both the host and bacteria, and then different rates of substrate. And we think especially oxygen and pH and so on is changed. And that's also what we see in the CF lung, because in the study I showed you before, we actually started to then think, so if we can find single cells in chronic infection, can we also find biofilms in acute pulmonary infections? So we investigated community-acquired pneumonia with as an acute infection, and then intermediate infection, the COPD patients, and a chronic infection, CF. And the remarkable thing is that we found the same amount of single cells and biofilms in all three infection categories. And there was also the same amount of inflammatory cells. But the difference was that the bacteria were growing much faster in the acute infections than in the chronic infections. Again, showing that, and this can also explain why acute infections are treatable with antibiotics and chronic infections are not. And we think this is due to the changed microenvironment. And we followed up on this also together with Marvin, where we have investigated CF sputum before, during, and after antibiotic treatment. And we can see, based on the amount of ribosomes in the bacteria as a proxy of growth, that after treatment, there's still a lot of bacteria present in the CF sputum, both single cells and biofilms, but they all have much reduced amount of ribosomes. So antibiotics can kill the metabolic active bacteria, but not the more dormant or inactive bacteria. So there's a changed metabolotype which cannot be eradicated by antibiotics. And we think that the metabolism of bacteria actually regulates the disease or infection trajectory. That if you have metabolic active bacteria, you have upregulation of inflammation and virulence factors, but they're also more susceptible to antibiotics. We have increased phagocytosis, and the disease trajectory is much worse. Whereas if we have the metabolic bacteria, then we probably have little less inflammation, or at least acute inflammation. We have less virulence factors, but we also see that the susceptibility goes down, and the disease trajectory becomes chronic and persistent, but it's not killing the bacteria and killing as much tissue as in acute infections. And we think there's this equilibrium between the activity of the bacteria, with the acute infections being dominated by metabolic active bacteria and the chronic infections by metabolic inactive bacteria. But then going away from the distinguishing between acute and chronic with planktonic and biofilms. And we think that a lot of the metabolism is probably controlled by signaling factors, for example, cyclic diGMP, oxygen, pH, and so on. But that is something that we are investigating these days. But try to put it into a perspective. We think that we try to coin now the bacteria metabolotype, linking the metabolic state together with infection persistence and antibiotic tolerance, and try to go away from not just focusing on the single bacteria and not just focusing on the lines in the case, but trying to understand the animals and bacteria in real life. As a conclusion, we think that I think that the biofilm paradigm remains a milestone, but not a destination. We have to move on, and I propose that the trajectory of infections is controlled by the metabolism of bacteria, not the aggregation of bacteria. And also that the amount of metabolic active bacteria is controlled by the infectious microenvironment, and the metabolotype of bacteria reflects metabolic state and readiness of bacteria in a certain environment. And that fits really well with another quote here from 1934, from some environmental microbiologists. They stated that everything is everywhere, but the environment selects. That's also that we find planktonic and biofilms everywhere, but the microenvironment selects the metabolism and the trajectory of the disease. So with that, all my fantastic collaborators, especially also Marvin and Frederik here today, and Phil Stewart, and of course also let's see, Bill Costerton, because I think what he did and the correlation he made has been a fantastic revolution within um, understanding bacteria in infections. But he would also agree that we need to move beyond this. We need to understand bacterial behavior in even greater depth. And he always believed that if you could change your um, uh, point of view, you could publish more papers also. So that is also very encouraging. And I think you would believe in this, that we cannot just stick with one notion just because it's been repeated over and over again. If it doesn't work, we have to investigate and dig deeper into it and find another explanation, another causes of these things. Um, and with that, I will hand over to Marvin, and you can start sharing, Marvin.
All right. Okay. Yeah, so today I'm going to hopefully build on what Thomas was talking about. There's not going to be a lot of data in this presentation, but it's really going to be just to give you a sense of the way we think about things, and hopefully it may make you think maybe a little bit differently, or maybe a lot of you think very similar to the way we think. But the idea is, uh, is in the title From Bedside to Bench. And it's instead of doing bench work to understand what happens in the clinic, maybe we should be in the clinic thinking about how this impacts what we do on the bench top. And, my lab works on a number of different infections. Most of these are chronic infections and coarse chronic wounds, which is why we're here today. But also we work on cystic fibrosis lung infections, and we also work on oral infections. And these are all really interesting infections. They have a lot of things in common. But today we're only going to focus on wounds. But I think one thing that's been really fun in my lab is to think about how an oral infection that may have 100 to 150 species in the infection actually, in a lot of ways, are quite similar to a chronic wound, which may only have 5 or 6 species in the infection. Most of what I'll tell you about today apply to 2 bacteria that you probably know a lot about, which is Pseudomonas aeruginosa and Staph aureus. These are 2 of the top 5 wound pathogens that we see in the clinic. And almost everything I'm going to tell you about today, the data mainly comes from working with these 2 organisms. So, what do we know about these types of infections, and whether this be CF or we know about our wounds, which is the 2 that we're going to focus on? Obviously, I'm going to focus more on wounds, but wounds and CF have a lot in common. The infections are polymicrobial. We know this is the case. Um, Sometimes you actually transition from a chronic infection to an acute infection. So there are times where you may have organisms, maybe they stay localized for a very long time and cause these chronic infections, but at times they can transition to acute. The other thing that we know is we can deliver really high concentrations of antimicrobials. Obviously, you guys know that you can deliver very high levels of antimicrobials to wounds, but you can also do this in CF, and that's why I have this here is the original antibiotics the original clinical trials that actually looked at inhaled tobramycin concentrations. Tobramycin's an aminoglycoside antibiotic, found that in the sputum in the lung, you could have over 1 milligram per mil of tobramycin. Of course, it's transient, doesn't stick around for very long, maybe an hour or 2, but you can get really high levels of antibiotics into these infections. The couple of things that we know is even though we can get really high levels of antibiotics in, first off, we often do not sterilize these infections. We do not kill all the bacteria there. Even though if we take these bacteria out of the lung or out of the wound and we grow them in the lab and we actually look with standard assays whether they're resistant to these antibiotics, this canonical antibiotic resistance is actually quite low, and it's a lot lower than you would predict it to actually be. So it's kind of interesting, right? You have these polymicrobial infections. You can get antibiotics in at levels that you ne almost never see resistance to, but you don't acquire lots of resistance and you don't sterilize these infections. And so that's the questions that we've really been thinking a lot about is why are we not sterilizing these infections? You can use hypochlorous acid, right, in wounds. You can get really high levels of antibiotics, but you're not sterilizing these infections. And then the other thing that my lab's quite interested in is why do these infections transition from chronic to acute? I'm going to give you some background on these and point you to some studies. Like I said, I'm not going to go through tons of data because I'd like to sort of let you think, mainly focus on how we think about these types of infections. But the question is, so these, I think these are really important questions, and these are a lot of labs work on these questions. And my lab early on was doing what most labs were doing. And how do you study these questions and how do we approach these questions? Well, you usually get a sample. Maybe you get a little bit of wound debridement, or you get a little bit of sputum from the cystic fibrosis lung, and you put it onto an agar plate. And oftentimes, what you get out is what you see here on the bottom left, right? You see, this is a plate that should grow mostly Pseudomonas aeruginosa. It's called Pseudomonas isolation agar. A lot of you know about this. There's a few other bugs that'll grow on this media, but what you see is you get all kinds of bacteria. You get big colonies, you get small colonies. If we zoomed in, you would see ones that have very different morphologies of the colonies. And then what we do is we usually pick one of these colonies. I mean, sometimes people pick a big one. Maybe they pick one that's a pretty color. Maybe you pick one that's small and kind of got an interesting kind of texture to it. And you take it and you streak it out and you get single colonies. This is what was done with PA01 and PA14 for Pseudomonas, the main strains that are used. It's also what was done for LAC, which is a big strain that was used in Staph aureus research. You take these individual strains and you put them in a test tube and you start doing experiments and you get these beautiful blue colors with Pseudomonas oftentimes, but not all isolates look blue. Some do, some don't, but maybe yours does. And you grow it in vitro in a test tube or you grow it in a biofilm model. Or maybe we take it and we put it on some human cells, right? These are human cells, and maybe you grow them in the lab and you put the bacteria on top. And you do experiments to ask how does the host maybe respond to the bacterium and how does the bacterium actually respond to the host? Then you may put them into insect models like Drosophila. You may put them into C. elegans, or you may put them into mice, right? And you do experiments. How fast does it kill a mouse? If I knock a gene out, does it still kill a mouse, right? What is the immune response of a mouse to this bacterium actually look at? And we've learned a lot doing these experiments, and these are the experiments that my lab has done for a really long time, over 20 years. And it was during this time as we were doing these experiments, and again, you can do co-infections, you can do genetics, we can image, we can do nanotechnology. We've done all of these things in these models. And then we sit back, and I really started to sit back and think, does this really matter? Are studying this single strain from this plate in these models, does it tell us something about the original infection that this sample actually came from? Does it tell us something about this that's valuable for understanding how bacteria behave in infection? And this was a question that we really thought a lot about. And the reason why I started worrying about this was a couple of things. One is what we found if we took a sample and we took a strain of Pseudomonas, let's say, out of a human, maybe from a human wound, and then we grew it in the lab. What we knew when we sequenced it was it very quickly acquire tens to hundreds of mutations when you passage it in the lab. And you guys all know this, right? If you call my lab and you get Pseudomonas aeruginosa PA01 strain from me and I send it to you, you will probably want to sequence it. And the reason is because my PA01 is not like Thomas's PA01. They're close, but they're not exactly the same. There's definitely going to be SNPs, and there's papers published on this. Bob Ernst's lab had a paper on this. So what we know from this is that when you take bacteria out of a human and you put it into an environment that it's never seen before, like lab media, it quickly acquires mutations and adapts to this. So this made us a little worried that, well, what does this tell us? Well, this tells us that maybe these lab environments are very different from a human, and maybe the behavior in these environments is not what we see in humans. The other thing that started happening is my lab started doing a lot of mouse models. And this was something that really was required, to be honest, to get funding, right? When you work with pathogens, they want to have mouse models. And we did mouse wound models. And it turns out that our wound model, you don't always get the same results in other wound models. And so maybe you make a mutation in Pseudomonas of a gene, and in our mouse model, we see that that gene is really critical for Pseudomonas to grow in that wound in the mouse. But then I use another mouse model and it doesn't. And so this really began to make us think, what are we learning from our models? It also made me start thinking differently, and I started going to a few different conferences because I really became convinced that I wasn't a molecular biologist anymore, although I'm a geneticist and molecular biologist. But what we were really studying was behavioral ecology. We were just studying behavioral ecology of bacteria. And this is very much like the behavioral ecologists have been studying for hundreds of years in larger animals, lizards and mammals and amphibians. And so how they actually do that kind of work is you go out and you observe and you measure and you assess and try to quantify the behavior of these animals. And the way you do that is you go out and you watch, right? And you record. And I started thinking, this is what we need to be doing for bacteria, and we need to be doing this in humans. We need to go out and we need to be doing behavioral ecology work and trying to understand what these bacteria are actually doing in their natural environment. And that be our benchmark for us trying to ask whether what we discover in the lab is relevant. So I use this a lot. I was very happy to see Thomas using lines and cages because I think I've used that one before. But I feel like this is for me. This slide actually came. I was actually in Oxford giving a talk, and I went to the pub with some graduate students who worked on were evolutionary biologists and ecologists, and they studied sort of larger animals. And this is really what it came that we sort of came up with that night, and we were thinking about it. And let's say that you didn't know anything about black bears, and you'd never heard of a black bear, you didn't know anything about it, and I gave you these two pieces of data. So both of these are observations that were made of black bears in nature, okay, out in the environment. Now, all of you know that this one here on the left, this is real data, right? You trust that this is real data because of what you know about black bears. But this data on the right here is just as real. This is an image that someone took of a black bear that just happened to be in someone's house. Now, if you didn't know anything about black bears and you got this image on the right-hand side, and this was the data that you were given, you might think that black bears lived in houses and they walked on 2 feet and they played the piano, right? These would be reasonable things that we would infer from this data, where obviously on the left we would think differently. So it really made me sort of think about how in the experiments that my lab was doing at the time, and I really thought that a lot of the experiments we were doing were giving us real data. Some of it was important, but I thought a lot of it may be studying black bears in houses. And so the environment really matters. And I really put this down here because it really matters for bacteria. So when you think about microbes, they respond very, very quickly to a new environment and very differently based on cues in that environment. And I want to give you a little bit of a little bit of data. I actually did this I was in grad school in the year 2000. Some of you probably weren't even born then, but I had this data. So what I did is I took Pseudomonas aeruginosa strain PA01. I grew it on an agar plate with a defined media with a single carbon source. And you can see I struck it here. You can see these lines. And what this is an antibiotic disc. So you guys have seen these before. It's called Kirby-Bauer test, and you put this antibiotic disc on. The antibiotic diffuses out, And if the bacteria are susceptible, they don't grow around the disc. And that's what you actually see here, this sort of black area here. These are 25-year-old pictures, so they're not the best. All I did is I just changed the carbon source. So I changed what I gave Pseudomonas to eat. And when I did that, this is the data you get. These are done on the same day. They've been repeated. They were done side by side. And all you have to do is change the carbon source, and you see the antibiotic no longer works. And you no longer see inhibition. So, and we can play these games all day. I can give you media that Pseudomonas will be susceptible to an antibiotic. I can tell you media where they're resistant to an antibiotic or tolerant to an antibiotic. I can give you media where they hyperproduce virulence factors, media where they don't. And because of this, it really made us begin to sit and think, without knowing what the environment is actually like, similar to what Thomas said, in the human, how do we understand and evaluate how good our data is that we get in the lab? But the question really becomes, how do you actually go out and do behavioral ecology in a wound, in a human wound? How do you understand what the bacteria are actually doing and what their functions actually are in these natural systems? So what we actually did was, and what we've decided to do to understand function is something called transcriptomics. And so what transcriptomics is a technique where we actually take a piece of a human wound out up from the wound. We take some debridement, maybe in CF, you take some sputum, and we actually extract the RNA from it, all the RNA. This would be human RNA, bacterial RNA, and we sequence the RNA. And then we focus in on the messenger RNAs for the bacteria. And using that data, we actually try to assess what the bacteria are actually doing. Now, most of you probably know that mRNA is correlated with protein, right? For the most part, it's correlated with protein. So the mRNA that we measure in these human infections tells us something about the proteins that are there. And the proteins give us the function. So from this data, we can actually understand, what are the bacteria eating? What are they? Are they stressed? What's their virulence factors look like? Are they producing a lot of virulence factors? And it's this data that we've actually been using. And the way this works is you might take a wound and you take a little piece of it out, again, like I said, and we immediately preserve it. This is, quite honestly, for any of the clinicians, clinical folks that are out there, this was one of the biggest issues that we actually had, was immediately preserving this. The average mRNA in Pseudomonas, the half-life is less than 3 minutes. So if you take a human sample and you set it on the bench for 10 minutes, while you're getting things together, there's been over 3 rounds of half-lives go through those RNAs. So you have to immediately preserve this. You takes it out and immediately preserve it. And this was our biggest problem, because the biggest challenge was just immediate preservation, right? And working with the clinicians, we've been able to do this. The other challenge is there's not a lot of bacteria in these infections. You guys think there are, but from a nucleic acid standpoint, there's very little. And there's difficulties in lysing the bacteria. But we've been able to get over this, and we've been able to assess the RNA transcriptome for Pseudomonas in these human infections. So from this, we've been able to ask a lot of questions. And one of the things is that, can we develop sort of models for wound research? Now, most of these models that I showed you at the beginning, when we develop models, it's most of the time through intuition. Like, I think most people would tell you that a mouse is better than a fly. They would say, well, a mouse has bones. It has an immune system that's closer to what we see in humans. And that intuition would tell us this. But what we've learned a lot as we've done transcriptomics is this is not necessarily the case. And so the way that we've actually done this is, instead of having intuition tell us that growing Pseudomonas on human cells is better than in a test tube, we've actually quantified this. And the way that we've done this is by looking at the transcriptome in humans and then looking at the transcriptome of Pseudomonas or staph when it's actually in our model. And then by just saying, how close are they? It can tell us where our models are good and where our models are not telling us something about humans. And we've spent a lot of time doing this and we developed a lot of models. The main work was done by these two postdocs who now have jobs. And here's some of the papers if you're interested. The other thing that I've recently done is founded a company called SynthBiome that actually develops models for wounds and CF and other infections that we provide for people so that they can grow their bacteria in a way that mimics what happens in humans. So the first thing that we've used this data for is to really develop these models so we can try to convince ourselves that using this model is valuable for understanding human infections. The other thing that we did is we've actually been able to learn new biology. And this was work by Pungbo Kao, who has his own lab now. And what Pungbo did is when he looked at Pseudomonas gene expression in human infections and in wounds, what he saw was that there was this one gene that was incredibly highly expressed in humans, but was during human infection, but not when we grew these bacteria in the lab. And it turns out that this was a small RNA and it actually controlled Pseudomonas, whether it sat as a chronic infection or it moved on to an acute infection. And it was this toggle switch that sort of told us about if it's highly expressed, it sits in a chronic infection. As expression wanes, it moves out and it's more of an acute infection. So just by looking at these and going out into nature and looking at transcription and looking at function, we were able to find some new biology. And there's a lot more of this. We found a lot more. It turns out small RNAs are also really differentially expressed. When we look at Staph aureus, when it grows in human infections, it expresses a lot of small RNAs that it almost never expresses in the lab. And we don't know the function of a lot of these because if something's not expressed in the lab, understanding its function is hard, right? Because you just don't normally see its function. And if you guys are interested, here's a review that we wrote with Kendra Rumball. And that's all I have. Thanks.
Thank you so much, Marvin. That was, as usual, really good presentation. And now we'll hear from Frederik, who will tell us maybe what to do and what not to do to use antibiotics and so on in chronic wounds.
So, I will try to not give you as in-depth presentation as Thomas and Marvin because I wouldn't be able to do that. But I'll try to illustrate what the impact of what Thomas and Marvin are doing actually is on the treatment of wounds from our perspectives. And I work as a clinical microbiologist in Denmark. And to those of you who don't know that, we, as clinical microbiologists in Denmark, are actually the ones in collaboration with with the infectious disease department who advise on treatment of bacterial infections. So we are involved on a daily basis in the clinical cases. And we, of course, are interested in the rational use of antibiotics because we know that antimicrobial resistance is currently one of the major threats to healthcare systems all around the world. And I have no conflicts here, but I do have some AI generated images, just that you know. Just a quick brush up because Thomas and Marvin has mentioned this already, but I just want to mention that when we look at the activity of antibiotic, we actually measure it in the concentrations of antibiotics that it inhibits the growth of a particular or specific bacteria, and this will be, of course, in the lab or in the wound or in serum or in whatever fluid you are using in it. Which means that one of the major parameters that we use is the concentration of antibiotics in relation to the MIC, which is the minimal inhibitory concentration. Again, the concentration. So actually, the effects of a particular antibiotic towards a particular bacteria depends very much of the drug concentration present in a specific area. This could be the wound, of course, a chronic or acute wound. And that's, of course, has a major impact on how we use antibiotics in a situation where we have either biofilms or dormant bacteria. And we also know that these parameters are all, I mean, how antibiotics are used, which dosage we should use, are all determined in the lab. They're all from in vitro systems. And the parameters or the variables that we use for the pharmacodynamics, they vary a little bit depending on which type of antibiotics we are talking about. If we talk about aminoglycosides such as tobramycin and gentamicin, the target or the goal for our concentration in a wound would be a concentration at the maximum concentration at one point should be 10 times the MIC. If we use ciprofloxacin, then we are aiming at an AUC, an area under the curve, MIC ratio of approximately 200, 400, 800 in that area. And for the beta-lactam antibiotics, the target for concentration of antibiotics would be that for the half of the time, within that means 12 hours of the day, the concentration needs to be 4 times the MIC. And that is, of course, a challenge if you're looking at difficult to assess areas such as abscesses, wounds, bones, teeth, whatever you're looking at. Now, we understand, of course, that we need to get our concentration of antibiotics to a certain level to have maximize efficacy or to reach the microbiological cure that we're looking for. But we're also looking into a concentration that cannot be too low. If the concentration gets too low, we might have a situation where there's a risk of resistance, antibiotic resistance is higher. We know that if you have sub-concentrations of antibiotics in certain areas, the risk of the bacteria becoming resistant is very high. So we have those two. We need to get high enough to have efficacy and also high enough to avoid resistance. But we are getting more and more aware of the risk of toxicity for using very high doses of antibiotics, especially with a population that is increasing in size and weight. And we know that now we're looking at this therapeutic window of antibiotic concentrations in patients so that we have like a minimal effective concentration for the desired response that we see, but we also have a top concentration of where we see adverse events, toxicity, bone marrow suppression, even some cognitive infusions in sense. So we know that we cannot just keep increasing the dose of antibiotics for patients that will cause toxicity. Now, of course, we also understand that when we're looking at antibiotic and resistance, the development of resistance is not just mutations in bacteria, as is very, very rare, but we do see another impact of selection of antibiotic resistance, which means that if you start treating patients with antibiotics, antibiotics will kill all the susceptible bacteria and leave the resistant bacteria behind. And this, of course, goes for throughout the body if you use systemic treatment. That can be for the gut flora, that can be for the skin flora, and of course it can also be for the wound flora, because we know that, and I will get back to that, wounds will of course be contaminated or colonized. And if you start treating patients with antibiotics, you will eradicate the susceptible strains such as the Staphylococci, and you will leave more difficult to treat bacteria such as Pseudomonas aeruginosa and Acinetobacter baumannii, which means that we are getting more and more concerned of the impact that not efficient antibiotic treatment can have on the population of bacteria in a specific site. And that, of course, also leads us to very much what Thomas says, that we understand, of course, that the way the antibiotics behave in vitro is very difficult and different from the way the bacteria act in vivo. And when we use our in vitro susceptibility testing, of course, the growth conditions are perfect for the bacteria. They have all the nutrition that they need, and we can see how antibiotics either inhibit or kill the strains that we're looking for. And then we, of course, can tell if the bacteria is susceptible or resistant. Now, of course, the resistant mark is important for us, because then we know it will not work to treat the antibiotics, whether you treat the bacteria, whatever you do. But if it's susceptible, we do now understand that susceptibility is not necessarily very much translatable from the in vitro susceptibility to the in vivo susceptibility, because as Thomas said, you have, of course, biofilms. The antibiotics cannot penetrate into where the bacteria is. We see, of course, host factors. We see in aquatic tissue. We see different things that will make it difficult for antibiotics to actually reach the target. Once they reach the target, you will have maybe a Pseudomonas aeruginosa or a Acinetobacter baumannii who's dormant, who's not actually in a proliferate state, and the antibiotics will not have the same impact as expected. Therefore, that's one of the issues that we're looking into when we try to avoid using antibiotics unless it's absolutely necessary for wound treatment, abscess treatment, etc. We also have, as I tried to mention before, a little bit of the infection continuum in wounds, which means that all wounds will be contaminated with bacteria. And when I talk about contamination, least this is bacteria that, from our point of view, does not proliferate and they do not interact with the host. We don't see any inflammation. The same can go for the colonization of bacteria. They can move from contamination to colonization. Now they're actually sticking around in the wound. They are proliferating. They are becoming more in numbers, higher in numbers, but we still do not see on interaction with the host. We don't see any inflammation. And sometimes we could say we will leave the colonization, we will leave the contamination. We don't necessarily need to treat, but this is a vigilant state. We need to make sure that colonization does not move into a local infection, because a local infection with the more readiness of the area, maybe you have some pus, maybe you have local changes in the skin, can of course move into spread around locally, becoming a large area that is red. You can see increased exudates, you can see increased redness, you can even see lymphangitis. And of course, this can spread into a systemic infection where you actually see either bacteremia or septicemia or deeper infection into the muscles and tissue below the wound. So, and of course, for local infection and for spreading of infections and for systemic infection, you need, of course, to intervene and use antibiotics. But we would, of course, say do not use antibiotics when you are in the contamination or colonization state, and use antibiotics when you are in the infection state. But of course, if it's only a local infection, perhaps topical antimicrobials or antiseptics will be the way to go before you go into the use of systemic antimicrobials. And to avoid going from the contamination colonization state to infection, we do need to embrace, or at least try to implement, the more non-antibiotics management strategies where we use very great or good wound hygiene. That means hygiene from the personnel treating the wounds. You also need to remove necrotic tissue. You need good cleaning of the wounds. You need the changing of the band-aids in a regular hour. Of course, you can flush the wounds with saline, but you can also use topical antiseptics. In Denmark, we use chlorhexidine, of course. We also use chlordine. You can use more toxic things such as hydrogen peroxide. You can even go into the use of silver. Of course, resistance to silver we do see that. Honey is mentioned in many of the international publications. We don't use honey a lot in Rigshospitalet, and it's not necessarily the honey that you buy in the supermarket you use. It has to be honey made for medical use. But those things you can use where you either use it as topical antiseptics, or you can use dressings that you have covered with different antimicrobial or antiseptics. You can also use local gentamicin, local colistin, but that is normally specialized treatment where you either have pharmacists preparing the solutions for you, or you have infectious disease specialists applying the solutions on the wounds. If you are in a very specialized hospital setting, such as Rigshospitalet, we use hyperbaric oxygen therapy for the larger chronic wounds. But this is more of a specialized treatment as well, where you really need to have special settings. You need to have specialists with understanding of how hyperbaric oxygen therapy should be performed. But those are the options that are non-antibiotic management strategies that you should consider before you apply antibiotics. And of course, if you then have to use antibiotics, and this is the case where you do see infection, I should say that it's very difficult. It can be very difficult, actually, to distinction between colonization and infection in the chronic wounds, also in the acute wounds, but especially in the chronic wounds. And when you have to use antibiotics, you have to go by the antibiotic stewardship ground rules, which is the right antibiotic treatment chosen for the right patient at the right dose and at the right duration. This means use the antibiotics that you know will have an impact on this particular wound. We'll get back to that. Use it for the right duration. Usually before the times, let's say a few years back, we would normally have given at least 2 weeks of antibiotic therapy. Now we're trying to decrease that to maybe say 5-7 days. And if no impact or only slow impact, either change treatment or prolong it for 10 days, 2 weeks, but that's the maximum. And of course, let's see if we can use antibiotics that are at least risk for adverse events for the patients, and of course, least risk for antimicrobial resistance to appear. Now, if you are choosing to use antibiotics as a clinical microbiologist, and as Thomas said, we still want the bacteria to be cultured on the conventional microbiology lab. We do need to see a swab from the cleaned wound. You clean the wound first and then you swab it. Do a thorough swab. Make sure you're not just culturing the colonization bacteria on top of the wound. You have to make sure you go into deep to the wound and get a swab, or if you can, get a biopsy to make sure we understand what's inside the wound, and then make us culture it. Let us make the antibiotic susceptibility testing to make sure that the antibiotics we apply are efficient, at least in vitro. And then once you decide to treat with antibiotics and you have the susceptibility testing, then we have to decide what type of antibiotics do we want to use. We, of course, have some bioavailable active stuff such as the rifampicin or fusidic acid, but we normally would advise not to use rifampicin or fusidic acid as monotherapy due to high levels of resistance. Currently, we're not advising on rifampicin and fusidic acid in combination, but use rifampicin with a beta-lactam antibiotics if you you want to use rifampicin. In Denmark, we use mesalinam a lot, not necessarily for wound treatment, but for urinary tract infections and such. And fosfomycin, which is like an old antibiotic, is now currently one of the last resort antibiotics. It's difficult to get your hands on in most countries, but it has a high level of penetration and could be efficient for many infections, also local infections. So I would say choose wisely when you have your antibiotics. Always ask your infectious disease specialist or clinical microbiologist how to treat, and the duration of treatment, of course, is important as well. But I think it's important to understand that once we start using antibiotics in a chronic wound with biofilm, with challenges of dormant or bacteria that are maybe tolerant for the antibiotics, it's very important that we understand that we do have an increased risk of antimicrobial resistance once we start treating, and we should look into alternative treatments before we choose the antibiotics. And we have to be completely honest, go back to the basics of wound care. We have to go back to the basics where we know that hygiene is the most important thing for wound care. We need that know that moisturing is important for wound care. If we can use debridement, remove the chronic wounds, the necrotic areas in the chronic wounds. Sorry, we can use local therapies, antiseptics before we go to antibiotics. We know it's more efficient because, as Thomas and Marvin has showed, treating bacteria in a chronic wound where the dormant state is present, where biofilm is present, is a challenge for all antibiotics. So antibiotics in treating wound care should be the last resort where we go to. I think those were the words for me.
Thank you so much, Frederik, and also Marvin. I think we have a few minutes for question, and there's three, as I can see it now. The first is from Emma. Hello, Emma. She writes, great presentation. How does the increased activity antibiotics in disaggregated bacteria align with the concept, probably the concept I presented? And I think it aligns really well because you will only see increased activity of antibiotics if you disaggregate the bacteria in nutrient-rich media. If you just disaggregate them in its own microenvironment, or the example in the bone or in the wound, you will not see increased activity. Only if you introduce oxygen or change the microenvironment. We actually have data on that. I didn't present it today. So I think it's one of the great misconceptions based on in vitro data that if you dissolve biofilms in fresh media, then they become susceptible. That's true, but this will not happen in a wound. Then we have another one. Maybe it could be for you. As mentioned, bacteria grow fast in acute wounds due to presence of metabolic active cells compared to chronic wounds. Does the antimicrobial treatment in both cases be different, and what will be the most common treatment option in both cases? Do you think?
No, but I think it's It's a very good question. Of course, as we talked about, the acute wounds where the bacteria grow fast, is likely that we see the more of, let's say, free living or plactotic state of the bacteria, and we would normally be quite efficient with using beta-lactam antibiotics, also narrow spectrum antibiotics such as oxacillin or penicillin. And if you then move into the chronic wounds, and the cells are more metabolically inactive, we are more likely to either use combination treatment in the case of Pseudomonas aeruginosa or Staphylococci, but we would probably also use antibiotics such as fluoroquinolone, ciprofloxacin, moxifloxacin that we know could be active on bacteria let's say, that are more metabolically inactive. So we would probably choose differently antibiotics depending on chronic or acute infections and wounds.
Mm. And there's one more question. So, growing skepsis about chlorhexidine, use drinking water, best approach, or should we start using other topical antimicrobials? And last week, I heard Klaus Møller give a presentation, and also with what Frederik just said, hygiene is the most important thing. So definitely not use drinking water because that is not sterile. And then I would not necessarily start using topical antimicrobials, but more treat the patients. If it's diabetic, make sure that the diabetic is controlled, offloading, compression, restore the vascularization of the limb, and so on and so on. To really start treating the patients and not just the bacteria, because a lot of wounds can heal even with bacteria present if the body is put into a better healing trajectory. Do you have anything to comment that, Frederik, Marvin?
No, I think this is the next question is actually good examples because we have to use topical antiseptics is sometimes the way that we go, especially also for the chronic wounds, because we know that for chronic wounds, systemic antibiotics can actually have difficulties treating the bacteria that we find in the wounds. And to avoid the continuous treatment of with systemic antibiotics, we try to use topical antiseptics to, let's say, keep the amount of bacteria to a minimum to avoid spreading of local infection, or at least to keep colonization there instead of infection. Let's say the application of local antibiotics such as gentamicin and colistin is normally something we do if we have our backs against the wall, where we don't really know how to proceed with this wound before surgery or during surgery. It is the let's say it's not the first choice. That'll be if we have no other choices.
Yeah. Then maybe last one. Increase immunity. I think treating the patient again, prehabilitation of patients before surgery, but also preventing wounds from getting infected, preventing breaches of skin, and finding the patients who are at risk and treating them very early. So I think it's also, as a community, we have to really work with first line practitioners and home care nurses. Wounds shouldn't be treated. Wounds should be treated much before they get into the wound clinics, because entering a wound clinic, I mean, then it's extremely difficult. So I don't think there's more. Marvin, do you have any roundup comments for the audience or anything?
I think it was really good. Yeah, it was great. It was fun.
Yeah. Cool. Then maybe there's one more, is a relation between using antiseptics and starting antibiotics. I think that, like, you probably just answered that, didn't you?
Yeah, but I mean, we try to use antiseptics without antibiotics, but in cases where we have difficulties getting rid of local infection or colonization bacteria, we can use antiseptics in combination with antibiotics, especially, as I think you said, in relation to surgical procedures.
Yeah. Yeah, I think then there's how does debridement of a wound not lead to another wound? I think again, changing the microenvironment is very good. Randy Walcott showed that you really have a window of opportunity when you change the microenvironment. Body can start to do its job again. And I think there's been a lot of evidence of resistance to silver in humans. Human wound model. I'm not 100% familiar with that is, but I think it is 6:06 now. Marvin is on holiday. He will eat turkeys in not too long. No, that will
That's
be
right.
later this weekend. I would like to thank everyone for attending. The webinar is on demand, and if you have any more questions or anything, you can write Julia at EWMA or get in contact with us. I think it has been an extremely good session. Thank you, Frederik and Marvin. And good night or good day to all of you out there. Thank you so much.
Thanks,
Thank
Thomas.
you.
Bye.